All right, so this is slides 14, and this will be the last chapter in this unit, unit 3. And this is the last one that will be included in this coming exam, which is exam 3. And it is kind of continuation of what we were talking about, which is a reproduction, but this is focusing more on what happened after fertilization of the egg. I will be again selective, and I will be focusing on the SLOs. Uh, and the first thing that we need to know is, generally speaking, when a sperm fertilize an egg. Remember, we talked about about twins before, so you do need to know that in a previous chapter. Fraternal versus identical twins. Is it one egg that's going to divide into two and produce two, which is identical twins? Those two will have the same genes. Fraternal twins, on the other hand, is two different eggs, so they will be different in genes and everything else. It just they will have the same age and they will grow at the same time and everything, but different genes because they come from different eggs. And what's going to happen is after that happen, fertilization, um, growth is going to happen, and growth is increase in size and also development, which is changing in a life phase to another. We will go from phase to phase to phase. Like the very first thing is fertilization of egg. This is day 1. Then what's going to happen after that? Day 2, 3, 4, 7, and going month, 2 month, 3 month until delivery of the baby. So what's going to happen here is we are going to divide the life phases into prenatal and postnatal. Natal means birth. Prenatal, pre is a prefix that means before. Post is a prefix that means after. So prenatal is before birth, which is basically pregnancy. Pregnancy is our focus. Postnatal is what happened during and after delivery of the baby. So we start again with prenatal, and this is from fertilization until we say it's 42 weeks, but it can be anywhere from 37 to 42. We usually say 9 months, but it doesn't have to be exactly like that. This is, again, usually we talk about averages. It's not like perfectly, it has to be like 42 or 40. No, it's 37 until 42. Less than 37 is called preterm. Pre-term, before term, meaning before full term. And if that's the case, the baby should stay in the incubator for some time because not everything is ready in that baby to live on his own, specifically something like the lung. The lung is not fully mature yet. Uh, he is alive and he will survive, and usually, and everything is okay, but he's not ready yet. At least his lung is not ready yet. Um, but when is this fetus viable? 25 weeks. Okay, this is about 6 months. Before that, he's not viable yet. So here is fertilization and how does it happen? And I talked about this before, but again, let's refresh our information. Remember that every month there will be follicles inside the ovary of females. And these follicles are going to grow under the effect of FSH, follicle stimulating hormone. And this growth of the follicle with the oocyte inside of it, this growth it will happen in 14 days, the first 14 days. At day 14 exactly, there will be a sudden surge of LH, luteinizing hormone. And the second oocyte specifically will be released. Then it will be picked up by these finger-like projections called fimbriae, and it will go to the infundibulum from which it will pass towards the uterus. I'm not saying it will go to the uterus. It moves toward the uterus. As it is moving, there will be a chance for a sperm to come from the other side, and not a sperm. I mean, the semen is here. A lot of these sperms will go up from the vagina to the cervix to the uterus and moving all the way until, if that the case, if there is sexual intercourse and if there is actually sperms, viable sperms moving up, and if that the case, if that happened, oops, sorry. Okay, if that the case and if that happened, usually it happened in this third, in this part right here. So this is going up, this is going down, I mean, and this is going up. They will meet somewhere around this area. We call it the upper third of the oviduct, or in the oviduct is good enough. During that time, if you still remember, we talked about the uterus and we mentioned endometrium, myometrium, and perimetrium. Remember the endometrium at that time, during the first 14 days or the half, while the follicle is growing, estrogen is making this wall thicker and rebuilding what was sloughed off during menstruation. I know I talked about that before, but again, I'm refreshing your information. So this is all happening during the first 14 days. The predominant hormone during that time that come from these follicles is estrogen. It go to the endometrium, rebuilding after it was lost. The sloughed off part of the endometrium is going to rebuild and it will become thicker and thicker. After the egg is released and being picked up, the remaining part, the remaining body, and body means corpus, it will change into a yellow body, corpus luteum. This corpus luteum is the one that's going to continue secreting estrogen, but it will add progesterone in the second half, the second 14 days. And this is to prepare this endometrium to become even thicker, but also secrete secretions. And these secretions are going to make it easy for the sperms to move up all the way until it meets, until it might meet the secondary oocytes. And if that's the case, fertilization occur. And this is the only time that you say it is a fully mature egg, meaning meiosis 2 will start creating polar bodies and zygotes. So we refreshed our information now. So now this is the fertilization. Still, I mentioned that. Look at this. There is a zone surrounding the secondary oocyte right here. And this zone, when one sperm moves in, penetrating it, and I talked about the acrosome and the digestive enzymes and everything. So once it releases the digestive enzymes from the acrosome and goes in, only the blue part, which is the nucleus containing the chromosomes, is going to merge or join with the secondary oocyte. At that time, something very interesting happened, is because of that joining, this zone surrounding the secondary oocyte, which is called zona pellucida, will become tougher and thicker and stronger, so that all these millions or hundreds of thousands, whatever the sperms that are available, will not be able to penetrate. So it will stop otherwise there will be like pregnancy with 100 or 10 or whatever. So once one enter, it's like you go in and close the door. So this zona pellucida, become thicker and will prevent the rest from coming in. They're trying, but they will not be able to make it. Just let's say this one, number 5, is the only one that were able to make it. And once it goes in, this zone become thicker and tougher. So look at all of these, they will not be able to make it. What will happen later is this fertilized egg will go into stages. The first one is called cleavage, and then embryonic stage, and then fetal stage. We used to confuse fetus with embryo. It is not the same. Fetus is not the same as embryo. Um, because embryo is the first part. Fetus is more when the embryo grow, it will become a fetus. So cleavage, embryo, fetus. So look at this. I'm not going to go through all the details. What I want you to know here is this egg coming, the sperm come from the other side, and then there will be changes of fertilization occur. Let's say fertilization occur. When fertilization occur, we call it day 0 at the time of fertilization. Now you have a zygote. And then you have the cleavage. Cleavage means breakdown. Like it's which is not really breakdown. It's like one cell, one fertilized egg become 2, 4, 8, and so on. So it go in multiple uh, cleavage, like day 1, day 2, like 2, 4, 8, and so on. And after that cleavage happen, we call it morula. Morula. Here is the morula. The morula after that, uh, like at day 6 or 7. So morula is like day 4. At day 6 or 7, the morula will become blastocyte, blastocyte, which will implant, meaning it will attach, implant, attach. And if you look at this, this is the blastocyte, this is the inner mass, and this is the outer mass. The outer mass is the one that's going to invade. We call that the trophoplast. And this will become the placenta. And this placenta is going to secrete something called HCG. And this is the base of pregnancy. Like if a lady wanted to know if she's pregnant or not, like she's not having the period, or even if she's just interested to know if she's pregnant or not, they just go to the pharmacy and get one of these pregnancy tests, and you can see if it is positive or negative. Basically, you're talking about this HCG. This is a hormone that's only formed if this trophoplast, which is external part of the blastocyte, invade into it and start to make the placenta. Uh, so if that's the case, what do you think? Like I'm saying blastocyte, which was morula, which was zygote. So what are we talking about? Definitely there is pregnancy. So this test basically detects this hormone that is secreted from this trophoplast when it invades into the mother's endometrium. And this is secreted because now you have pregnancy. And this is going to support the pregnancy. So definitely, this is the one um, that can be detected. For your information, that's kind of too much, but for your information, it appears early during the first few days in blood. And then later it will start to appear in the urine. So the most frequently used one is the urine one. So the urine one is kind of a little bit late, not very late, like after a week or something, a week or more when it starts. But actually, it's even more than that because this will happen during like 6 or 7. So this is trophoplast invading after that. So like day 7, 8, something you can detect in the blood, and then it will accumulate and start to appear in the urine later on. Let's say it's 2 weeks now or something. This is kind of details that you don't need to know, just if you are interested to know. Next is you will have an embryo. And this embryo is, there is an inner part surrounded by fluid, and then there is an outer part. The inner part is called embryonic disc. The outer part is called chorion. In between, there is amniotic cavity containing amniotic fluid that's going to form. It actually is called the extra embryonic cavity. And this part here is the one that's called amnion, uh, which is basically, I want you to know that the embryo will be suspended in the amniotic fluid. The embryo, which is inside of this, will be surrounded by fluid. Good enough to know. It will start to divide into different divisions and will start to make the different body parts. You can read it if you're interested, but just understand the idea. Here, you will have the fetus surrounded by amniotic fluid. And there will be a connection here to the endometrium of the mom. This is the endometrium of the mom. So in between those, this part here will be the placenta, and this part will be the umbilical cord connecting the baby to the placenta, which is partly from the mom's endometrium and partly from the fetus. Just I want you to understand something here. At this time, they can insert a needle into this fluid here and get a sample. We call that amniotic fluid sample. And look at these chorionic villi, these villi here, which will make the placenta. They can also take a sample of that. So those two samples can help us predict if this baby is going to be normal or not. We do genetic testing. We call it prenatal genetic testing. Remember, prenatal means during pregnancy. Postnatal means after birth. Remember, natal means birth. Pre means before. So can we do that? Yes, they can get insert a needle and get a sample of this, or they can get a sample from the chorionic villi around it here. Here is the chorion. So these villi, they can just go in here without going into the fetus inside. You just take it from here or from here, which is chorionic villi versus amniotic fluid. Then the fetus, I mean the embryo, it will continue to grow until week 8. After week 8, he's not an embryo anymore. We call it fetus. So what's the difference between embryo and fetus? Embryo is until week 8, until he's 2 months old. After that, it is not. We call it a fetus. The fetal will have this fetal circulation. He will be receiving blood from the mom, oxygenated blood with nutrient from the mom, and he will be sending his waste products and carbon dioxide back to the mom. See, who's dependent on his mom. So is he breathing? Of course not. The first breath is when the baby cry after birth. Before that, how can, like, we breathe to get oxygen and get rid of carbon dioxide? Yes. How can he do that? Through the placenta and through the umbilical cord. There will be umbilical artery and umbilical vein. Umbilical artery is the blue one that will take deoxygenated blood and waste products to the mom. And the umbilical vein is the red one that takes oxygen and nutrients to the fetus. The fetus, again, will continue to grow until like 38 weeks. It's ready to be born. And the way it will be born, look at this cervix here. This cervix will start to dilate, and look at the head of the baby. Will start to push in, and basically, when the cervix start to dilate, it will secrete. It will elicit or promote the secretion of oxytocin, and oxytocin will go and contract the myometrium or the uterine muscles, which will push the baby out. The heads will fit more into the cervix. The cervix dilate more, secrete more oxytocin, more contraction, more pushing to the baby, more dilation of the cervix, more Translation, positive feedback. Positive feedback. And this part is important to know. So look at this. The baby will go out. We're not going to go into more details than that. What I want you to know here is how the process work. It's positive feedback. It's oxytocin. There will be also some prostaglandin that will contract the muscles. So prostaglandin contract the muscle, pushing the baby before oxytocin start to kick in. One more thing, just for your information, not in this, not in the basics, just for your information. The head of the baby goes out. This is the majority, but not necessarily every single delivery. It doesn't have to be like this. Sometimes it's called breech, like the hips is the one that fits in and start first. This is unusual. Is it an issue? Not an issue, it's just harder. Sometimes it is the shoulder, sometimes it's even the hand start first. There are different things that you are not responsible to know. Just normally it is the head. And then when the baby goes out, we have to cut the placenta. Once you cut the placenta, no more oxygen go to the baby, and carbon dioxide does not leave the baby anymore. So the baby start to breathe. And this is where the baby, we say the baby is crying. He started to breathe because oxygen drops. And this will send the signal to the brain. We are not getting oxygen anymore from the mom. They cut the umbilical cord. We're not receiving anymore. Oxygen goes down, carbon dioxide goes up. That send the signal to the brain, start breathing. Okay. So this is basically what we need to know. I know I skipped a lot, which is what I told you is more than enough. That's more than enough to know. So this is what we talked about: growth, increased size, development. Is it changing in the different phases, which are prenatal and postnatal? Prenatal, before birth, which is from fertilization until birth. Postnatal is from birth until death, like the baby is born and started to grow until death. Prenatal is cleavage, embryo, and fetus. I told you that. Cleavage is the first part, first couple of days. And then it will become embryo after attachment of the morula to the endometrium and started to make the placenta and the umbilical cord until week 8. After week 8, we call it fetus. During the first few days, I know that I said that the cells multiply, but does not increase in size. Like this is the size that contain 2 cells, same size but 4 cells, same size but 8 cells, until it become morula. Later on, to start to grow. Uh, is the whole entire blood of the mom go to the baby and the whole blood of the baby go to the mom? No, that's not true. There is a filter that allow in the placenta that allow just the good things to go, like oxygen and nutrients, to pass from the mother's blood to the baby or to the fetus or the embryo, and allowing carbon dioxide and waste products to leave the fetal blood and go to the mother's blood. That's it. If there is anything else, it's not allowed because there is a barrier, a filter. I'm not going to go into more details than that. So after birth, the mom will start to have her milk. The very first milk is called colostrum. It's kind of yellowish a little bit in the beginning, but the baby will take it. And I just want you to remember that this process here is also positive feedback. We talked about that before, which is the baby is suckling on the nipples. And because of that, oxytocin is released, which will go to the muscles surrounding the glands in the mother's breast, or the mammary glands, and it's going to squeeze it, pushing the milk out. The baby is sucking more, it's releasing more oxytocin, contracting more, pushing more milk out. So remember that this is a positive feedback. This milk is extremely important because it contains a lot of antibodies, especially in the beginning, because the newborn does not have his immune system mature yet. So he gets ready to use antibodies. So the baby now is breathing on his own and his organs are working. It will be divided postnatal into infancy, childhood, adolescence, adulthood, senescence, which is sen coming from senility, and adult is coming from uh, teenage, or yes, like these from 10 until like whatever 15, 16. So sen is coming from senility, and then until the end of life. And you can read the details if you guys are interested in that, but this is kind of beyond our scope. So just know that infancy is until 1 year, childhood is from 1 year until puberty, and puberty is different from person to person. And then adolescence is from puberty to adulthood. Adulthood is from adolescence to old age. And then old age is, which is called senescence, is until death. Of course, everybody's different. Some people survive and live until 100 or 120 or whatever. Some people can die earlier and so on, which is again beyond our scope. So this is what's happening during each one of those periods. We start with uh, neonatal period, and then we go to infancy, childhood, adolescence, adulthood, senescence. Here are two more slides or three more slides that are important to know. It is related to what we talked about, which is in vivo versus in vitro. These are not in your slides, but I replaced the slides. So use those three slides. Um, it is in our SLOs, but it was not covered much in a slide, so I added those. So if you already downloaded the old version without those three slides, just download it again or add those three slides, whatever you want. It's nothing much, just something that that's we need to know, which is we talked already about pregnancy. Are there alternatives if this couple are not able to to make conception and do fertilization? What can we do? There are two things here. There's in vivo and in vitro. Vivo means inside the mother's uterus, inside. Vitro means outside, in the lab. So in vivo is they take a sperm from the male, the dad, and they inject it into the oviduct, which is a fallopian tube, during the time of ovulation, so that it fertilizes the egg. And this will be used only if we know for sure that this person is okay, his sperms are okay, but for whatever reason, they cannot go all the way up until the fallopian tubes. They tried and tried and tried, and it didn't work out. So what's going to happen here is, okay, maybe there is an issue in the uterus that preventing that. So we are going to bypass this pathway, which is from the vagina to the cervix to the uterus to the fallopian tube. We're going to bypass all of this. We are going to inject directly into the fallopian tubes. And then fertilization will occur just as normal, and it's going to move down all what we talked about. Uh, fertilized egg, and then the day 1 until day 6, and all these trophoplast and morula, fetus, I mean embryo, and then fetus, and so on. Everything else will happen. But the bottom line is inject directly into the fallopian tube. The other thing is, if this was not the reason, it's not just interruption. Um, there is another thing that if they failed, even that failed, and or if they know that there is no issue that's affecting the sperms to go all the way to the fetus. It's actually maybe the mom is having a big issue. So if that's the case, they do something called in vitro. In vitro means in the laboratory, outside, which is procedures um, that can be used in this case, or sometimes it's used to prevent genetic problems. Like if they know for sure that those two couples, if they tried to do like conception, fertilization, there is a good chance to get genetic abnormalities. If that's the case, they can get several sperms from the male, and they can get a couple of eggs from the female and grow them at the same time. They grow separately in a dish. It's a laboratory dish. Or they make it grow like in the laboratory somehow, and then they study them and they see which is the best sperm that does not have genetic issues, that's that have all the vitality, it's healthy, everything is good, and get this egg that's good egg that does not have issues, and so on. They grow, and then they put them together and they fertilize in a dish, not in the fallopian tube. Whether it's because this lady cannot conceive, she's having an issue, whatever it is, there are a lot of issues. I'm not going to go into the details unless if you guys want to know, but it's not in our scope. Basically, if she's having an issue preventing her from normal pregnancy, what can we do now? Get not one sperm, get group sperms, get group of eggs, examine those, get the best, put it in a dish. They add hormones that was supposed to be added. The mom should be adding. They will add it from outside until it grow outside. So in vitro means outside, in vivo means inside. The other thing that I want you to know is during the prenatal period, pregnant ladies should go to the doctor to do certain things to make sure that everything is good. Otherwise, if something is missed, it might lead to issues. So one of those is ultrasound during pregnancy. Ultrasound is one of the safe uh, tools to give a picture of the baby. It's usually done around that time, 18 to 20 weeks. So let's say 4 to 5 months. Sometimes it's done before that, but this is the average. And this is not an X-ray for your information. Pregnant lady should not do X-ray. X-ray can be harmful. No X-ray. Ultrasound is the safest to the baby and to the mom. And it will give a picture and show us what's happening in there. The size of the baby, is it normal size? Is it big? Is it small? If there is any issue that can cause birth defects, there is an issue with genes, there is an issue with the vertebral column, the brain, like I mean the skull, and so on. It can give us a good picture um, to see what's happening in there and decide accordingly. The last thing that I want you to know here is the prenatal genetic test. So prenatal is not ultrasound. It's not only blood test. There is also genetic test. So what can be done if the lady is pregnant? Ultrasound, blood tests, prenatal genetic tests can be done. It's an option. Automatically, ultrasound and blood test, automatically, like ABC. Once you go, yes, I'm here for prenatal care. Okay, let's start with ultrasound and blood test, and then go from there. These are like the bottom line, basics. Blood tests would check like if there is vitamin deficiencies, if there is something unusual in the blood, something is high, something is low, and so on. Too much details, just know the idea. What do we actually do? Ultrasound, blood tests. Another option is prenatal genetic tests. You know what's prenatal now. There are two types of those. I talked about it. Chorionic villus sampling. You get a sample from the chorionic villi. Remember, I showed you, you insert the needle. The chorion is the outside. You can get a sample of these villi, or amniocentesis. What's amniocentesis? Amnio is the amniotic fluid that I showed you, the blue fluids around the baby. And synthesis means a sample with a needle. That's what it means. And they can diagnose for genetic diseases. Is that something routine? No, it is not. It is not routine. It's something that can be requested. When would anybody request? Anybody can request it, but it's usually for if there's something expected. Like we did genetics, and you know, like the homozygous and heterozygous, and the chances of inheriting something. So, what are the chances of having sickle cell disease? What are the chances of this? So they usually are not done routinely upon request, and more likely if there is the genetic analysis say there is a chance for something. And we did, if you remember, the Punnett square and the pedigree and all that stuff. So the reason for that is to give the parents information about the fetus, their fetus, what to expect. If the fetus is having this issue, what should you do? And what will be the needs? And do you want to actually proceed, or you want to end the pregnancy or something like that? It will give you expectations and uh, whether you want to end the pregnancy or not. I'm not going to discuss any details related to anything else other than physiology and anatomy. From that genetic analysis, they can know the sex of the baby. They can see if the baby is having something like Down syndrome or neural tube defect. Neural tube is this vertebral column that contain the spinal cord. Some fetuses babies or fetuses can have like a defect, some sort of a defect, a bone that's missing or something, and at least they can take care of it. And they can tell you it will have a Down syndrome and so on. Give you the options. So those three slides are also important, and I covered all what we need to know for the purpose of this course, and I even expanded beyond that a little bit, whatever is related to it, more than enough. So as usual, this is repeating what we talked about already: growth, development, prenatal, postnatal, positive feedback during child pregnancy and during breastfeeding. We briefly talked about the different stages, and we don't need to know the details, just know how it goes from neonatal, infancy, childhood, adolescent, adulthood, and senescence. So this is basically what we need to know with this chapter. I'm going to stop sharing and stop recording.